Okay, we're looking at DNA in this chapter 13, DNA and RNA, primarily DNA, and some of the characteristics of DNA, and about DNA replication, and some of the characteristics of RNA, the process of making RNA, which is transcription and then a translation, and some things that cause genetic mutations. So we just finished talking about our studying about populations and how populations change over time. And a lot of that is due to environmental pressures. And so when we look at environmental pressures, occasionally we see mutations that happen at the genetic level. And we'll look at some causes of that. So in the duplication of DNA, sometimes mistakes are made, and we'll try and identify some of those potential possibilities in the replication of DNA. Some of the essential questions that we want to look at in this chapter would be, what are the rules for base pairing? Base pairing is, DNA has certain chemicals in it, and they're paired always the same. We also want to look at how errors are made during replication of DNA, the differences in the structure of DNA and RNA, and also, why is DNA important? One of the early scientists that looked at DNA and what was going on. Bacteria are really a simple organism to look at. They don't have very many DNA base pairs, and so it's less complex to look at bacterial DNA. And so in 1928, Griffith experimented with pneumococcus bacteria. The word coccus means round. So bacteria are classified according to their shape. Sometimes they're spiral shaped, sometimes this round shaped or rod shaped. And then they can also be classified by size, not just shape, size. And then whether they infect plants or animals. But he experimented with this pneumococcus bacteria. There's a link on here to a video about Griffith's experiment that shows you a little more in detail about what he did. It's not very long. I think it's maybe 4 or 5 minutes, but it's pretty good. So if you get a chance, take a look at it. But Griffith experimented with the bacteria that causes pneumonia. And there's 2 forms. One, the S form is deadly and the R form is not deadly. So to prove this, he took some of the deadly bacteria, injected it into a healthy mouse, and the mouse died. And then he took the deadly bacteria, the R, and injected it into a mouse, and it survived. Next, he did and he didn't call it DNA because it didn't have a name at that time, but he said there was some kind of transforming material in organisms, and to prove that and that they could pass from one organism to another, he took the deadly bacteria and he heat killed it. So that's the same reason that we cook ground beef. We're cooking it, raising the temperature, because then it will kill the bacteria, which would keep us from getting sick. So to prove that, he took this S bacteria that was deadly. He heat killed it. He injected it into the little mouse, and the mouse didn't die. So, but then the idea was, okay, now I've got this harmless bacteria here. Let's mix it with this harmless bacteria, living harmless bacteria. And so when he injected the mixture into the mouse, it died. And it thought a long time why this was, and wasn't really sure about the results. And it took other scientists looking at the same experiment to kind of puzzle out what the answer was. If many times when scientists or even medical, you get the swab in the back of your throat to grow the bacteria that's in your, maybe causing strep, that's what they're looking for. They're looking at the size and the shape of the bacteria. So in looking at the two forms of pneumococcus bacteria on these agar plates that are used to grow bacteria, you could see the differences in the two different bacterias. After they were heat killed, when he tried to grow this deadly bacteria, he heat killed it, there was no growth. So that proved that all those bacteria were dead. When he mixed the two, the harmless here and the heat killed, and injected it, and the mouse died, he took a blood sample of the mouse and he found this disease-causing bacteria, much like what was in the original. So in thinking about that for a long time, and other scientists, Avery said that it was DNA that was the transforming principle that changed Griffith's pneumococcus bacteria from a harmless bacteria to a deadly. So this, and he performed a lot of tests on this, the DNA of the two, and he, to show that DNA was present. DNA is made up of, it's a chemical compound, so it's made up of nitrogen and phosphorus bases. And then looking at the samples, he found that there were the percentages and the ratios of nitrogen to phosphorus were the same. So this is a known value for DNA, and that was comparable to what was being found in bacteria. So this further helped to establish that DNA was that transforming principle in the bacteria in Griffith's experiment. A couple of other scientists that came along was Hershey and Chase. They did some experiments with viruses. So now if we think about viruses are a little bit different. We don't classify them. They're not organisms, so they're not living things. Viruses are classified based on what they infect. If they infect bacteria, plants, animals, both. Also, their size. Some of them are very small. Some of them are larger. They're much smaller than bacteria. But then the other thing that's used to classify them or identify them is based on, do they have DNA or do they have RNA? So some of your viruses, so virals, DNA can be tagged, and the way they do that is because DNA has phosphorus in it. They can tag it with radioactive phosphorus in order to track the DNA as it infects one cell to the next. So in order for a virus to reproduce, it can't reproduce on its own because it's not a living thing. Remember, to be living, what do you want to do? You want to survive, do your thing, and then reproduce. So collect food, survive, and reproduce. So bacteria cannot reproduce on their own. So they hijack a cell, whether it's bacteria or plant or animal cell. Viruses will inject their DNA into them in order to make more of the viruses. And that can be a problem in getting rid of viruses because once a virus is in your cell, your body doesn't recognize it as being foreign because it's made its DNA part of your DNA for that cell. So it's a little problematic in getting rid of viruses. So in Hershey Chase's experiment, that's what they did, was they tagged the radioactive phosphorus in the DNA, and the DNA of the bacteriophages, these they're called bacteriophages, but they're viruses that infect bacteria. So this virus here, excuse me, they tag the DNA with radioactive phosphorus. And then in the other, they tag the protein code, which is not genetic material, with a radioactive sulfur. When they follow the infection of a bacteria, they found the radioactive DNA inside the bacteria, incorporated with the bacteria's DNA. So the phosphorus that was originally in the bacteria's DNA had incorporated that DNA from the virus because it had the radioactive phosphorus present. Here, there wasn't any radioactivity noted in the DNA of the bacteria because the bacteria doesn't incorporate the protein coat, which is, a non-nucleic acid. But anyway, this experiment further proved that, viruses can transform cells by injecting their DNA into an organism's, the host or DNA. This has become very useful because this is actually how they do gene therapy. So if you have some DNA in a person that, has a mutation and something's not working, you can actually insert a good section of DNA into a cell. For a long time, you could figure out where a mistake was in the DNA, but how do you fix it? Because DNA is so small, you can't just cut it out, put something. So viruses have become a delivery vehicle for good sections or corrected non-mutated sections of DNA in the cells. Down here, if this video link, if you will look at, this is how the AIDS virus is used as a delivery mechanism, because AIDS viruses can get into cells very easily. So in delivering a good copy of genes or genetics to a cell, and that's what this video link is about. And so it's about 10 minutes long, but this is for a deadly childhood disorder. But using this gene therapy, it has been able to keep children alive that have this mutation because the DNA is corrected by inserting a good copy. So DNA is composed of 4 different types of nucleotides. The nucleotides are monomers units. Mono means one, that make up the DNA. So the 3 parts of the DNA are the phosphate group. So you have a phosphate group here, deoxyribose. How do I know this is a sugar? Because it ends in OSE. Remember, glucose, lactose, maltose, sucrose, cellulose, all those oses, those are sugars. And then this nitrogen base, so this part, the nitrogen base can be either adenine, cytosine, guanine, or thymine. So those are the 4 principal bases in all DNA, whether it's plant DNA or animal DNA or even bacteria DNA. It only has these 4 bases, and they pair in, there's a definite pairing between the 2. 2 of the bases, the purines, are adenine and guanine, and the 2 pyrimidines are cytosine and thymine. Now the way that I remember these is pyrimidine, it has this Y in it, and so does cytosine and thymine. So thymine and cytosine are single ring structures, and here's your single ring. So it's a carbon ring, and here's your single ring. So thymine looks like this, but instead of drawing all these elements and bonds and things, you just see this little shape here. So thymine and cytosine are single rings, and then adenine and guanine are double rings. And then looking at the pairing, cytosine bonds with guanine, and thymine will bond with adenine. This is because if I put a double ring here and a double ring here, then the distance in my rungs of my ladder would be too long. And if I put a single and a single together, if I put C's and a T together, then you would have different lengths in the middle. So if we flatten this out to be a ladder, the shape of it would not be consistent. So you're getting a long double molecule and a single, and then a long double, and then a shorter single. So lengthwise, all of these rungs of our DNA ladder are going to be consistent. So nucleic acids can be either DNA, deoxyribonucleic acid, or ribonucleic acid. So they control the cell growth and reproduction. Again, A's only bond with T's, and C's and G's, guanine and cytosine and adenine and thymine. A good way that I always tell students to remember this, you can think of apples are in trees and cars go in garages. So cars in garages and apples in trees. A's and T's and C's and G's. We'll get to a little difference in the RNA, but not much. So the deoxyribonucleic acid, there's your D, R, N, A. Oh, well, ribonucleic acid, D, N, and the A. It's also called the code of Y. It determines those alleles or those characteristics or the phenotype, what we express or what we see, the characteristics of an organism. And there's some of those recessive genes that we may carry traits or alleles that aren't expressed unless there's two recessive together. But Cardos has some rules about, the adenine, apples only bond with thymine, the trees, and cytosine and guanine. So these two always bond together, A's and T's and C's and G's. So anytime that they pair differently, this will be a mistake or a mutation. And sometimes that happens is there's a substitution for maybe a G here because you have a double instead of what should be an A. Even bacteria, if we look at E. coli, you've got the A's and T's and C's and G's. And this is a flattened out, the strand of DNA, it's a spiral double helix, but flattening it out like this makes it a little bit easier to see the pairings. But even looking at the percentages, whether it's bacteria or plants or animals, you're still getting the same percentages and the same ratio of the phosphorus and the nitrogen bases. Be sure, so this is just a little introductory right now. Be sure to go to Jolleyology, the website, and you can check for things that you need to complete in this chapter, the assignments, there's a list. And then also be sure to do your vocabulary practice for your test so that there's a lot of probably new material in this, so you can get a head start by looking over that and not getting behind maybe or getting overwhelmed with too much vocabulary at once. But if you start now and do it a little bit every day, that